Thank you very much, Barry. What Barry didn't mention was that I co-direct the Clinical Scholars Program, and he directs it. So I work very closely with him as well. Well, thank you all very much for coming out on this chilly night. And I'm honored to be here to continue the Science 101 series of the Parents in Science Initiative. And tonight we're going to be discussing immunology. And it's really Immunology 101 through the lens of vaccinology, or an ounce of prevention, vaccines in your child's immune system. So I can't tell you how happy it makes me to be standing at this podium, because I first came here a very long time ago. This is my ID from 1979. Note that I had to scrub out the Social Security number, which appeared as my employee number at the time. I had one of these in 1978 and 1977. Those were the first summers that I worked here. I couldn't find them. This one I still have. Notice the slight difference. I'm not sure you would recognize me. I'm not sure you can recognize me here either, but I had the great privilege of coming to work with, as Barry mentioned, Dr. Ralph Steinman and his mentor, Dr. Zanville Cohen. And for those of you who can't see me and Ralph in the picture, there we are. Obviously, quite different. And Dr. Cohen's in there as well as several of my friends and colleagues who are still here. Maggie Pack and Svetlana Mozov are in that picture as well. This is Dr. Cohen and Ralph together working on likely a dendritic cell paper. And this is a photograph from their original paper in the Journal of Experimental Medicine in 1974. The first one appeared in 1973, describing the tree-like cells, or dendritic cells, that they identified that are the orchestrators of immunity. And they have been the sort of central focus of all of my scientific work, and they are also the central focus of all vaccines that work. So you'll be hearing a little bit more about them as I proceed, but you might keep this image in mind. After many years of struggling hard to convince the scientific community that these cells first existed and how important they were, Ralph was awarded the Nobel Prize in 2001 in Physiology and Medicine for his role of the discovery of dendritic cells and its role in adaptive immunity. As many of you know, sadly, he was not able to accept the prize as he passed away 3 days before the prize was awarded. I had the great privilege of accompanying his wife Claudia and his family to receive that prize almost exactly 2 years ago. So, with that said, we're going to move on to the vaccine, to Immunology 101, or the properties of the immune system. So the immune system, unlike other systems in the body, doesn't have a specific location. It's throughout the body, at all body surfaces, including the skin and every organ. And it both has positive and negative effects. So there's a switch that goes both ways. So you have immunity, or the establishment of protection, vaccine protection, or protection from a disease once you have it and don't get it again, like mumps, measles, or rubella. And then also, as many of you have followed recently, there have been great advances in cancer immune therapy, or the ability to manipulate the immune system to fight off cancer, both to prevent it through vaccines like the vaccine for hepatitis B and for human papillomavirus, as well as to fight established tumors through intentional manipulation of immunity. It's also responsible for autoimmunity, and that includes diseases like lupus and rheumatoid arthritis. Lupus was defined here by Henry Kunkel. Using a very small number of patients in our hospital, he really defined this disease in a way that we still rely upon. And type 1 diabetes is another form of autoimmune disease. And these diseases and the others like them are good targets for immune therapy of various sorts. Much of the earlier therapy for them was sort of like taking a sledgehammer, using steroids to quiet the immune system. But in these diseases, the body's ability to not make a response to its own self is somehow disrupted. The third general category of diseases that, again, is of great interest, I think, to all of us, especially those of us who are parents, is allergy. And that is where the body makes an overactive response to a foreign antigen that it would usually be tolerant to. And these include asthma or respiratory allergy, food allergy, which, as we all know, has just overcome the world, or our world anyway, and also atopic dermatitis. Unfortunately, I only have, or fortunately, depending how you look at it, I only have half an hour to talk. And if I had all the time in the world, I would be able to go on at great length about all of these things, but I'm not going to. One of my friends and colleagues asked me how my lecture was coming last week, and I said it was great, except it was 2 hours long. And I promise you it's not 2 hours long now. But if I were to talk about these things, it would be at least 2 hours long. So these are topics for another day, and I hope that we'll have the opportunity to address them. But they're important, and the immune system relates to them. So to understand immunity, there are a certain number of sort of vocabulary words that I'd like to introduce to you. For those people who are familiar with them, forgive me, but I just want to make sure we're all on the same page. So these are the sort of essential elements in immunity. The first is an antigen, and that is any substance, either foreign or self, that is recognized by the immune system. So an antigen can be as diverse as a microorganism, a bacteria or a virus, or pollen in terms of allergy, vaccines, or even one's own cells. So in cases like lupus or rheumatoid arthritis, the antigen is actually some component of the person who's mounting the immune response's own body. An antibody is the protein that's made in the blood that is made by B-cell lymphocytes, and this is known as the humoral arm of the immune system. This is not cell-associated and can be easily transferred from one person to another. Cellular immunity, which is the response of the cells of the immune system, and these cells directly respond to the antigen. So B cells make a substance that binds to the antigen. T cells are the cellular arm of immunity. The cells directly target it themselves. So in terms of the orchestrated immune response, there are certain characteristics that characterize it and are very important. And they are specificity. So an immune response to the flu virus does nothing to the rhinovirus that causes colds. So the immune response is specific. There's also great diversity. So if you were to travel to Africa and encounter a whole bunch of new microorganisms, your body would be able to respond. And that's diversity. And perhaps the most important characteristic we talk about is memory. And that means that when you got measles, mumps, rubella, or chickenpox as a child, you made an immune response. And when your child gets it and you're re-exposed, you don't get sick because your body remembers. Your immune system has memory. And that's one of the most important characteristics of it. So these three characteristics of specificity, diversity, and memory characterize the immune response. So in making the immune response, there are B-cell lymphocytes, which I mentioned earlier in association with antibody. And these are the antibody-producing cells. They're called B cells because they were first identified in birds that have a bursa, which is a sort of common organ in birds, but not in mammals. And they produce immunoglobulin, or IgG, in response to antigen. This is a cartoon of what antibody looks like. It has a Y shape. And this is important later on, you'll see why. And there's a light chain and a heavy chain that bind together. And this is called the variable region. So this will be specific, say for flu, or for adenovirus, or for your tumor cell. And this is the constant region, called the FC portion, which remains constant. These may be neutralizing, which means that they kill something, or they may be non-neutralizing, which means that they just bind. T cell lymphocytes, which are effector cells, they're the cells that kill by directly touching. They come from an organ called the thymus, which is an immune organ that sits directly above the heart. The thymus is the largest it ever is in a human being at the age of 2. And by the time you're 7, it's all over. It involutes, and only the cells are left. So CD4 T cells are helper cells, and CD8 cells kill. They are critical for antiviral and anti-tumor immunity particularly. And now we get to, again, the cell of interest in my life called dendritic cells. These are antigen-presenting cells that pick up antigen, process it, which means they shuffle it around in a way that the other cells can see it, and then they present it to the other cells in a way that it can be seen upon their surface. And this is the site of, these are the anatomic sites of the immune system and also of dendritic cells. So you have lymph nodes in all of these sites, and in the tonsils and adenoids, which are in your mouth and nose, as well as on the skin and all mucosal surfaces. These are the sites that generate both immunity and tolerance. So it's a little bit hard to sort of conceptualize, unlike, say, the kidneys, which are right here, they are not in a particular place, but rather spread throughout the body. So when I first started working with Ralph, back when I looked like that in the other pictures, he had just described dendritic cells, and my first scientific lectures were basically to convince the people to whom I was speaking that dendritic cells both existed and they had a unique function. And then the next sort of set of scientific lectures, we had a great slide that he had made that had dendritic cells as the commanders and all the other cells as the soldiers. Well, I spent a decade working at the Walter Reed Army Institute of Research, and I had the opportunity to brief the general 1 or 2 times. Well, briefing the general was a very big deal, and frankly, most of the time they weren't interested. But when I was able to show, I should probably not say that, but that was the case. But when I was able to show this slide that had, you know, this sort of martial theme to it, and the idea of the commanders and the soldiers, all of a sudden they all perked up. They were very interested, and I got their attention. And so I loved that slide. But Ralph felt that our understanding of dendritic cells as a family of cells, where there are many, and of all of the responding cells, of which there are many, the analogy was much better to call them the conductors and the immune orchestra. So this is the elegance and scope of immunology, cells of the immune orchestra. During Ralph's lifetime, I rather stubbornly kept my Marshall version of this slide in my slide deck, but with his passing, I figured I better do what he had said. And so I took instruction, and that's his choice. So this is a beautiful picture of dendritic cells, courtesy of our friends and colleagues Nikki Romani and Gerald Schuller in Germany. And these are human dendritic cells that are derived from blood monocytes in a phased microscopic image. So I think you can see their dendritic processes. And Ralph, when he first identified these cells, this is really a lot like what he saw in the microscope. And his lovely wife, Claudia, who is with us today someplace, has very long arms and legs. And he wanted to call them Claudiocytes. And he apparently came home, and Claudia can elaborate on this, and she convinced him otherwise. But she would accompany him to meetings, and people would look at her to sort of scope out the extent of her extremities. But so these are dendritic cells in all their glory. And this is a cartoon of a live dendritic cell in a mouse doing what it does, which is finding a T cell and presenting antigen to it. And this is courtesy of my colleague, Michelle Nussenzweig. So this is a loop, and I could watch the loop all evening, but it just started again, and I won't make you watch it all evening. So this whole beautiful orchestra of immunity is what we rely on to protect us from disease. So vaccines are an attempt by us as physician scientists to basically fool the body into thinking that it has seen the disease, mounting an immune response, so the next time it sees the disease, you don't get sick, you rather eliminate it via all of the mechanisms that I just mentioned. So one of the things that's really hard, it's a little bit like global warming, I suppose, or security. When something doesn't happen, you don't notice. So vaccines protect very, very well. My grandmother was a physician, and she graduated from medical school in 1921. And she used to like to describe in great detail to me children dying from diphtheria. Diphtheria caused a respiratory disease that caused choking, and it was epidemic in New York, and it was horrible because children would choke to death. And one of the things I think that inspired me to do the work I do was her description. And then her description, because as I said, she graduated from medical school in 1921, of what happened after 1920 when the vaccine was introduced. So if you look at 1920, this is how many cases there were in the US, and in 2009 there were 0. I don't think there are very many physicians working today, including myself, who would even recognize diphtheria if we saw it. Maybe we would. We'd hope we would. But we wouldn't have recognized it from having seen it commonly. And I'm not going to go through all of these and share them with you, but you can see basically the point of this is vaccines work. And the best example of vaccines working is smallpox, which is the only disease which has been actually eradicated from the face of the earth by the use of vaccines. So the idea that vaccines work has been one that has been near and dear to my heart for a long time. And I was just delighted when the New England Journal of Medicine arrived, and then it was quoted in the New York Times the next day. And Barry mentioned this in his introduction, that Don Burke and his team at the University of Pittsburgh did a huge meta-analysis, taking all of this public health data that they could get their hands on to demonstrate that vaccine programs for children have prevented more than 100 million cases of serious contagious disease in the United States since 1924. And the way they analyzed the data and portrayed it, and I could show you in much more detail, but I, again, will control myself, but this is a heat map. And I think even from the audience, you can see this is measles, and they did this for all of the vaccine-preventable diseases. You can see the year that the vaccine was introduced, and the darker colors are fewer cases, the lighter colors are more. And you can see how the disease is decreasing. So they looked at 56 diseases. And, the report covers 56 diseases, but focuses on 7, and all 7 have these beautiful heat maps that you can look at later, or I can show you. I have in my slide deck at the end if you're interested. So the whole principle of this is what does not kill you makes you stronger. And this principle, actually the word vaccine itself comes from the word vaca, or cow. And that arises from Edward Jenner, who was an English country physician who knew neither of viruses or immune cells. And in fact, in his lifetime, new and sort of controversial science was Harvey's idea of the circulation of the blood. So it's just kind of amazing to think in of him and how in the environment in which he worked. But he noticed, he made good observations, and this is at the core of all science and certainly all clinical science, that he noticed that milkmaids had very smooth skin. Smallpox gives you pock marks. Milkmaids had very smooth skin. They were known for that. And they also didn't get smallpox. However, they had a single sore on their hands often, which they seemed to get from milking the cow. And this was a lesion of cowpox. So he had the idea that somehow this lesion was protecting them from the smallpox infection. So what he did was he took, and pardon me if anybody's got a, is squeamish, he took the crusts of these sores from the milkmaids and he scratched them using these ivory points. Remember, there are no hypodermic needles at this point, into the skin, first of orphan children and then of his own family. Now, clearly, this would not pass the IRB, the Institutional Review, at all. I sit on the IRB, and let me tell you, not a chance. But this vaccine, which was developed in 1796, went on virtually unchanged to eradicate smallpox from the face of the earth. And at this point, it exists only in labs here at the CDC, and it was in Russia, and who knows where those frozen samples are now. But it no longer circulates. And in fact, in 1972 in the United States, smallpox vaccination was stopped because the risk of the vaccination exceeded the risk of contracting a disease that had been eradicated. So though I am acclaiming the wonderful role that vaccines have played, after the smallpox vaccine was developed, there were very few vaccines that were in common use, and yet diphtheria and tetanus were terrible diseases and killed many, many people, as we saw from that earlier slide. So Dr. Kitsasatu and Emil von Behring, separately in the 1890s, developed antisera, which demonstrated the protective role of antibodies. So antibody-based therapy against diphtheria in humans was developed in 1894. And you can see here, this is the US mortality from diphtheria. This is the introduction of the vaccine. But it was already decreasing significantly here because of the use of antitoxin. And so the idea of antitoxin was not to vaccinate somebody and have them make their own immune response, rather to vaccinate a horse and have them make an immune response and purify the antiserum, or what we now know to be the immunoglobulin, and then administer that to the person. And it actually works remarkably well. The problems with it are not that it doesn't work, it's that the human makes an immune response to the horse, and upon a second administration, you can get a very serious disease called serum sickness from the immune response to the horse serum, which is what this was. My physician grandmother, who liked to tell me about diphtheria, worked on antiserum therapy for pneumonia. So that was actually developed and perfected here in our little hospital, which if you turn around when you leave and look to the right, you'll be able to see. It looks lovely tonight, but not nearly as lovely as it does in the spring. And at the hospital, in the first decades of this institution's existence, there was a huge breakthrough was the development of treating pneumonia with antiserum of horses. And in fact, when you drive on the FDR, if you look to the left, if you're going north, or to the right if you're going south, you can see the large arches through which the horses were brought on a barge from the very special farm they had in Princeton where they kept them, and they fed them special food and kept them very clean. I don't exactly know what they did, but they used to bring them in those doors, and it's still there. I don't think they open anymore. But Louis Pasteur changed, so though antisera was useful, there were limitations, particularly because of the nature of the horse serum. Vaccine science matured, largely thanks to Louis Pasteur, who Jenner made good observations, and he took advantage of an experiment of nature. But Pasteur was a real microbiologist, and he identified microbes and then he attenuated them, which meant he made them weaker in such a way that they didn't cause disease but would induce immunity, and he used them to produce vaccines. He famously produced the rabies vaccine. Max Tyler, who worked in Founders Hall, produced the yellow fever vaccine, and the polio vaccine was produced by Enders Weller, based on technology developed by Enders Weller and Robbins, was developed by Salk and later by Sabin. So I could again tell you great stories about all of those vaccines, but I won't. But for those of you who are interested, there are many references that I could provide to read about them. But I will tell you a little bit about the polio vaccine. I don't know how many of you remember before there was a polio vaccine, but this poor little boy is in what's called an iron lung because infantile paralysis, his respiratory muscles were paralyzed, and this nurse is caring for him. This is an electron micrograph of the polio virus. So the polio was, as some of you remember and others of you who are younger don't, a terrible, terrible scourge. Children were kept inside in the summertime. They couldn't go swimming. And it was really, it was dreadful. And then Salk and then, as I said, Sabin developed a vaccine. And really, I don't believe anybody expected it to work, except perhaps Dr. Salk, but it did. And this is the card. There was a large field trial conducted, which school children were vaccinated. Again, I don't believe it would pass the IRB these days, but it was conducted in what was considered to be an ethical manner at the time. And this is the card, and this is the button these children were given. And I don't believe that I'm violating HIPAA because I have permission to share that Dr. Coller actually participated in the early development as a volunteer, not as a physician, as a little child, in the development of this vaccine. Sadly, even though polio, like smallpox, could be eradicated from the face of the earth because it has no intermediate host. So if a microorganism has an intermediate host, and that's an animal or an insect, it's very hard to eradicate because how do you get it out of that reservoir? But for illnesses like smallpox and polio that don't, eradication is possible. And the technology exists to eradicate it globally, but social and political issues have stood in the way. And these poor children in West Africa have suffered infantile paralysis, and you can see they have braces on their legs. In 2007 and 2008, there were only a few cases left in South Sudan, and it looked like polio eradication was just over the horizon. And sadly, due to a variety of political and complex social problems, polio is now spread to 17 countries, I believe, including Syria. Again, I feel as though I'm like advertising for the New York Times here, but I'm always so excited when there are vaccine articles that I can present to you. And this was, again, just from recently in November, that because of the war in Syria, they've stopped vaccinating. And even though vaccines work beautifully, they only work when you use them. And this has been demonstrated over and over again. When we lose our vigilance and we don't administer them regularly, diseases break out, and this is a very good example. So another vaccine-preventable disease that is in the news is meningitis, the meningitis vaccine. So the meningitis vaccine was developed by Dr. Emil Gottschalk, who is on the faculty here and chairs our IRB. And this is a photograph of him from 1978 when he was awarded the Lasker Award for the invention and development of the meningococcal vaccine. So the meningococcal vaccine that he developed is for meningitis A, and meningitis B is not covered by that vaccine. So many of you may have followed that meningitis B has had an outbreak at Princeton, and I believe at another university in California, the name of which escapes me for the moment. And so they have pursued an IND, an investigational new drug application with the FDA, to allow the use of a vaccine that's been approved in Australia and in Europe, which works very well to prevent meningitis B, and that's called Bexsero. So I believe that I heard on the radio that the children at Princeton are being provided this vaccine now under this special sort of dispensation. So now to get to, for those of you who are parents in science or grandparents, the CDC, I'm not giving any medical advice to anybody. I am a great believer in vaccines, and as I like to say about Dr. Gottschalk, he's the only person I know who made a vaccine that's in my children. There are many other vaccines in my children. I just don't know the inventors of those vaccines so well. But these are the CDC recommendations for the vaccines. And this is for infants and children, and this is for teenagers. And the thing about this is this changes. There's a group of people, many of whom I know and some of whom are here, who go to the CDC on an annual basis and assemble these recommendations. And they change based on new vaccines that are developed, new formulations that come out, and frankly, how diseases change in the population. So I think it's very important to follow these and to follow the advice of your pediatrician, who is probably getting these updates on a regular basis. So these are the vaccines that are recommended for adults, and the two that I'd like to bring your attention to, or the three, forgive me, that I'd like to bring your attention to, are the influenza vaccine, which everybody should get, the new DPT vaccine, which has been reformulated in a much less reactogenic formulation. So it's now being recommended particularly for those adults who are around small children. And then the human papillomavirus vaccine. This is a wonderful vaccine because it actually prevents cancer. So a number of epithelial cancers, including cervical cancer, are caused by the human papillomavirus, and this vaccine prevents them. These ages refer only to the ages for which the vaccine is approved, because those are the ages in which the studies were conducted, but studies for older people are underway. Again, these are catch-up childhood vaccines, and then there are a whole host of other vaccines if one is traveling. Hepatitis A and B, influenza, everybody should probably get. Again, the meningococcal vaccine as well as others. So this is just a chart of the CDC recommendations for vaccination 0 to 18. So you can see it's fairly complicated and fairly intense. So I would just recommend the website and following the advice of your pediatrician. So I feel as though I have special authority to discuss childhood vaccination because I'm actually the mother of 5 children and the stepmother of another 5. So yeah, I've got more boys than anybody in the room, I bet you. So my children don't actually like me to show their picture. None of them are here tonight, so I can do it with license. But these are my children and my mother, who is here tonight, and my husband, who's also here. So as I said, vaccines only work when you use them. And unfortunately, they've, as Barry alluded to, they've become highly controversial for a whole series of reasons, including the idea that somehow vaccines were associated with autism. And there were a series of publications that really, in the British Medical Journal, that really caused great concern among many parents. Unfortunately, or fortunately, these publications, these scientific publications, it's been concluded that they were in fact the result of an elaborate fraud. And the data was so flawed that it's not just wrong, but it was fraudulent. So many people, particularly in England, have resisted vaccinating their children. And as I said, when you don't vaccinate, the diseases come back. So there have been several outbreaks of measles and mumps and rubella as well. So I think that this is both reassuring in that this link is not authentic, and a reminder that we have to be vigilant in vaccinating. So those are vaccines that work, and they're wonderful, and they work when you use them, and there are social problems that get in the way. And there were 100,000 cases of measles in West Africa last year. There is a measles vaccine series that costs $8 per child and works beautifully. The problem is getting it to the children who need it. One of the things that I was delighted to hear is the Gates Foundation has now embraced this as what they call low-hanging fruit, and they are, in fact, paying for this to occur. There are still challenges in doing that, but there are infectious diseases that resist vaccine protection. And these are diseases that are chronic, not acute infections, and they're diseases where getting them and getting better does not confer natural protection. So the whole idea of the smallpox vaccine was that you would get it, and your body would think you had smallpox, you'd get better, and then you'd be left with long-term durable immunity. Those are the hallmarks of good protection. And for tuberculosis, malaria, and HIV, natural infection does not confer those attributes. So as Ralph liked to say, with these we have to be smarter than nature. And my work in the last 2 decades has been focused on an HIV vaccine, but these principles extend to these other challenges as well. Incidentally, the immune challenges of these vaccines are probably very close to the immune challenges of cancer immune therapy. Many of the problems are the same. So just to remind you that HIV is a tremendous problem, even though due to the highly effective antiretroviral therapy that has been developed, it's much, much better here in the developed world. There were still 7,000 new infections every day in 2012. 97% of these infections are in low and middle income countries, and 1,000 are in children under 15 years of age. About 6,000 are in adults 15 years of age or older, 41% are young people, and 51% are women. So to make a new vaccine or a new drug, it has to go through the approval process of the relevant regulatory authority. In the United States, that's the US FDA. So we work under their authority and we work closely with them. So this process has 3 phases. There's actually a 4th phase, which is post-licensure, but until something gets licensed and can be sold, there are 3 phases. Phase 1, which is safety. And I cannot emphasize enough that safety is paramount. I have several different slides that say like safety, 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 safety. And I thought I would spare you that. But in all of our work and in all vaccine development, the Hippocratic principle of first do no harm is preeminent. The 2nd phase is immunogenicity, and that's showing that the vaccine induces the immune response that you think you're going to need. And I emphasize think. And the 3rd phase, and only the 3rd phase, is efficacy. And that's where you go into a large field trial, like I mentioned earlier with the polio field trial, and administer the vaccine to a large group of people who are at risk, and you administer a placebo to a similar group, and then you compare who got infected and who was protected. And it's only when efficacy is established that a vaccine will be licensed. So all of the studies that I'm going to tell you about are phase 1 very early safety studies, where we're collecting a little bit of phase 2 immunogenicity data. So once again, for those of you who didn't look at this is a dendritic cell, and this is an image again from Ralph and Dr. Cohen's original publication. And I put this here just to remind you that when you go call a plumber, you get plumbing. So when you ask a dendritic cell biologist to design a vaccine, you get a dendritic cell targeting vaccine, which I think is a very good thing, because remember, dendritic cells orchestrate immunity. So all vaccines that work actually go through dendritic cells, even though nobody knew they existed when many of them were developed. So Ralph's idea was that if we could directly target a dendritic cell, if we could get the vaccine to where it needed to be, maybe we could establish a kind of immunity that was better than natural infection and would be protective. So we have just finished the conduct of a study which was conducted by my colleague Marina Kasky of a randomized double-blind placebo-controlled phase 1 study to evaluate the safety and immunogenicity of our dendritic cell targeting vaccine and an adjuvant, which is something that helps it work better, in healthy volunteers. And if you recall, I showed earlier that an antibody is shaped like a Y, and so we have an antibody that knows how to find dendritic cells, and on its end, an HIV vaccine. And the idea is that this antibody will bring this vaccine directly to dendritic cells. So this was an idea in Ralph's head in 2001 when I came back here to work with him to bring his ideas in the clinic. And it has now been developed as a candidate. So anything that isn't licensed is called a candidate and has been in 45 human subjects. This is our IND. This is the paperwork that we had to submit to the FDA to get permission to start our trial. Now, with our next trial that I'm going to tell you about, we did that electronically, so I don't get to show you all the pages, but this gives you an idea of the amount of work that goes into starting the trial and also that gets reviewed to make sure that these things are safe and appropriate for testing in humans. These are the results, and I really don't expect you to understand them, nor will I interpret them for you, but suffice it to say, we've completed the trial, and we've demonstrated that this very special vaccine strategy produces a moderately good immune response, both in the humoral compartment, which is antibody, and in the cellular compartment. But what's most interesting to us is this humoral response, which we think is very important based on other colleagues' vaccine studies, seems to last a long time. So we're pursuing this finding. So our current work, which even though it makes me very excited to talk about immunology in general, but it makes me most excited to talk about the work that we're doing now, is with my friend and colleague, Michel Nussenzweig. Michel is a head of lab here. He was one of Ralph's first graduate students, and he now heads a lab focused primarily on antibodies. So if you recall, earlier in the talk, I mentioned the diphtheria antitoxin, and it works really well, except for the fact that you get antibodies to the horse serum in which it's raised. So Michel has figured a way to make antibodies that are made synthetically, so they're not raised in horses, but they're highly effective and highly neutralizing to HIV. So our purpose is to have a small phase 1 trial to evaluate the safety and the ability of these best-in-class monoclonal, that's the term for these particular special antibodies, that are broad and potent in humans. And we just literally got our communication from the FDA today, moving that trial forward into the clinic in the beginning of 2014. The site of all of these studies is our lovely little hospital, which is the first. This institution was founded in 2001, and the second building that was opened was the hospital in 2010. We celebrated their centennial in 2010. We are currently supported by the Center for Clinical and Translational Science, was created in 2006. It's one of the first 12 NIH-funded CTSAs, and Dr. Coller is the PI of this grant, as well as being our Vice President for Medical Affairs. Is that the right title? Our hospital is fully accredited and has dedicated inpatient and outpatient units, and only, as Mr. Carson said in the video, only sees research patients. So we do not generate a bill, which is one of the things that actually makes me very proud to work there. So in closing, I'd like to, since this is a Parents in Science enterprise, I would guess many of you, if not most of you, have been to Central Park, not too far from here on East 67th Street, and seen this picture of Balto. And I don't know that many of you would know, but this statue of Balto, the sled dog, has to do with passive immune therapy, which is what we're working on now. So he carried, I'm going to actually read from the inscription. This is dedicated to the indomitable spirit of the sled dogs who relayed the antitoxin, 600 vials, across treacherous waters through the blizzard and he went from Anchorage to the relief of the stricken children of Nome in the winter of 1935. So endurance, fidelity, and intelligence. So the course of the Iditarod is based on this transport of diphtheria antitoxin from Anchorage to Nome to save the children of Nome, which it did. And so I feel like we've got a long heritage of developing passive immune therapy along with our heritage of developing vaccines. And it makes me very happy when I take my children to the park and I see Balto. So with that, I'd like to thank those who've made this research possible. We are funded by the Bill and Melinda Gates Foundation, through the foundation, through NIH, the National Institute of Allergy and Infectious Diseases. We've received tremendous internal funding and support, especially when we needed it most. All of our studies are supported generously by the Center for Clinical and Translational Sciences, which I mentioned earlier. We also received some funding from the Robert Mapplethorpe Foundation, and we've been supported by more than 150 contributors to the Zan Cohen and Ralph Steinman Professorship, which is held now by Michelle Nussenzweig. And this is a beautiful picture of the university at sunset. So thank you all for your attention. And I'll be happy to entertain questions.
So now, after this great presentation, we have an opportunity for you in the audience to ask your questions.
Thank you for that talk. You mentioned adjuvants a little bit in there, and I've heard that aluminum is used as one of the adjuvants, and that's a little controversial. I'm curious if that's accurate or not.
Right, actually, it's not aluminum, it's alum. So it's not related to aluminum, just by name. So it's a different compound. It is somewhat there are other adjuvants that are quite controversial, but alum is actually the adjuvant that's been used the longest, and I believe the only one that's actually approved by the FDA currently for use in this country and is very safe. Aluminum obviously has many problems with it. It's been associated with dementia and Alzheimer's disease. I don't know how strong those associations are, but alum is not related to it in any way.
You want to maybe just say a word or two about what an adjuvant is?
Oh, absolutely. So an adjuvant is a compound that's added to a vaccine to help it work better. So one of the things that's of great makes me very happy is when I started in this field, people talked about adjuvants as though they were like some mysterious potion, and they sort of regarded them that way. In fact, they work by stimulating dendritic cells. And there's been a tremendous amount of work by our group and by many, many other groups across the world demonstrating how each of these adjuvants very beautifully molecularly stimulates different populations of dendritic cells as well as other immune stimulatory cells. But alum is, so there are now what are called molecular adjuvants, which are very specific and target one pathway or another in a very cognate manner. Alum is a more general adjuvant and has a long history of use. I think it was probably first used in the 30s and is generally held to be very safe. It only works for protein vaccines. So it doesn't work for DNA vaccines or viral vectored vaccines. There are all different kinds of ways to make a vaccine. It only works for protein. And it works in one way by sort of aggregating the proteins in a way that make them more appealing to the cells to pick up. And I believe it directly signals through one of the TLRs, but I don't remember which one, so I don't want to misspeak.
That was a lovely talk. Thanks very much.
Thank you, Mary.
My question has to do with vaccine efficacy and why some seem to work better than others, and some, there are, for example, in the hepatitis B vaccine, not everybody is a responder. And there's that question. And the second is why some vaccines work, why we lose our capacity for a immune response as we get older.
Well, so that you've asked several very good questions. So the first is vaccines, all vaccines are imperfect in one way or another. And depending on the vaccine strategy, there's a balance between safety, reactogenicity, and durability of response. So the smallpox vaccine, the one that Edward Jenner invented, which I've received, and you actually still give, I know. So if I might, Mary is what I like to refer to as our school nurse. she's director of occupational health, and she is the person who gives me my flu shots and gives many of us our vaccines when we travel. And so she's very knowledgeable about their use. And because there are people here who work with viruses that are related to smallpox in the laboratory, this is one of the few places where one might actually have a smallpox vaccine still administered. And it's administered in a different way. It's administered directly into the skin with a little needle. And it's very, very potent. Immunity to smallpox vaccine likely lasts a lifetime. But as you know, it's a highly reactogenic vaccine. It makes people sick. You can get really sick. You get a big swollen lymph node. And a certain number of people, I believe there was, so vaccination stopped in 1972. There was great worry after 9/11 that there was going to be a weaponization of smallpox. And there was vaccination of a large group of first responders with this very, very potent but highly reactogenic vaccine. And I believe 12 people got seriously ill, and 7 actually died as a result of the vaccine. So if you vaccinate 1 million people with the smallpox vaccine, depending on the study, between 1 and 3 will die of vaccine-related complications. So if you're facing a smallpox epidemic where 30% of the people are going to die from the illness, that's a pretty good rate of, that's a good cost-benefit analysis. But if you've got a disease that's eradicated and you're talking about a potential threat and a sure mortality, the calculus, so there's a lot of controversy, a lot of discussion. The decision was not to vaccinate. So that's a very, very, very effective vaccine, not so safe as vaccines go. The flu vaccine, which is extremely safe, immunity probably only lasts a year. That's why we need to get constantly revaccinated. And that's not a terribly good vaccine. There are a whole lot of reasons why that one is still used, but there are many people who are making efforts to make better, more durable flu vaccines that cover more different strains. So there's the basic nature of the vaccine, and then there's also the issue with the flu where the organism against which you're protecting mutates. And so if you have a vaccine strain and the vaccine's not that good, it will only protect against something very close to it if the organism has changed and gotten farther away from it, which microorganisms tend to do. Then you'll still have protection, but it won't work against the circulating strain. And the reason that it's harder to induce immunity in older people is there's what's called senescence of the immune system. And we start to lose the ability to make robust responses probably right after puberty. It starts to decrease, and then it really decreases around 35. And then once you hit 50, it's all downhill. So. Yeah. And there's great effort in trying to figure out how to overcome that, adjuvants that might be used, because it is harder to vaccinate people, as you know, for flu once they get older to get a good response, and they're the people who most need protection. So that's a hot area of research.
There was a phase when people were inoculated with the BCG vaccine against tuberculosis. That doesn't seem to happen anymore.
So again, it's a very good question. So the question was, there was a time when people were inoculated with BCG, which is Bacille Camille-Guérin. Pardon my bad French accent. But it's an attenuated bacteria that's related to tuberculosis but doesn't cause disease and gives some immunity. So it's still, I believe, used in some countries in the developing world because we have issues with it with testing for TB. There was an analysis done, and I'm not actually familiar with the details of the analysis, in the probably 60s, that demonstrated in this country the use of the time test, so the ability to do a skin test to see if one had been exposed and then treat. So if you have not been vaccinated with BCG and you get a time test, a little tiny bit of antigen is injected beneath your skin, you don't make a response and you're negative, and it's clear you've not been exposed to tuberculosis. If you've been exposed and you have a time test, you make a little lump. A whole bunch of T cells come to that antigen site, and then that's called a positive test. Another thing that Mary does for all of us. And I'm sure probably everybody in the room has had one. If you have a positive test, that indicates in this country that you've been exposed to the bacteria, you've been exposed to tuberculosis, and you were then treated. So tuberculosis is a fascinating illness because most people who are exposed to it don't get infected, and most people who get infected don't get sick. But it's very hard to predict who those people are going to be. Though there are individuals engaged in doing genetic studies trying to do that, for now we don't know. So what the public health strategy in this country has been is to detect people who've been exposed and treat them early on in their lives, and then that prevents them from getting sick later on. If you vaccinate with BCG, you make a response. You demonstrate immunity. So you no longer can distinguish people on that basis. And it's not a terribly good vaccine. I think the protection rate is only in 70-something percent. And to be licensed in this country, generally speaking, and I use the word generally speaking, the FDA wants to see greater than 90% protection. So, but it still is used some places in the developing world where they don't have the ability to do this testing, and the analysis has been that it's better to protect 70% of the people than nobody at all.
I guess you know that vaccines have become kind of controversial, and parents are concerned about negative effects.
Absolutely.
And I was wondering, and as we all know, drugs can go on the market and then later off be taken off the market.
And that has happened with some vaccines as well.
And that happened with DTaP. So how do we know, what is the research that you are doing or have done or somebody else has done to see what are the worst effects that could possibly happen to even one child in getting over 15 different vaccinations at a very young age? They're now tons, I mean, every month.
Right, no, believe me, I know, and I have children. One of the things that has happened that I actually feel very good about is that the vaccines used to have what's called thimerosal, which was a preservative in them to prevent any kind of contamination from growing, and that was mercury-based. And even though there were all these calculations that showed, you know, even with all 15 vaccines, it was a safe amount, my feeling was there wasn't a safe amount. I mean, there was no good reason to be giving heavy metals to small children. And the FDA has agreed with that, and now in the US, that preservative is no longer used. So that's one of the things that's been done to demonstrate protection. What you're asking, and you know, as a mother, I share this with you, you know, that you want to know that this isn't going to hurt even one child. Unfortunately, there's no guarantee of that with anything. You know, there are risks in everything that we do. There are risks in what we do, and there are risks in what we don't do. There are risks in crossing First Avenue. You can get hit by a car. And if you stay in bed, you know, like the astronauts up in space, and you don't get up and walk, your bones will disintegrate. You know, I mean, I realize that these are extremes. There are some risks anytime you encounter a needle. You know, I mean, when you draw blood, we have consents for drawing blood that have all of the potential risks for having a standard blood draw. You can get a bruise, you can faint and hit your head, you know, on and on. But there are very, very small risks, and the benefits outweigh the risks. With licensed vaccines, my view is, certainly with the vaccine preventable diseases, that the benefits so outweigh the risks, which they really are very safe. What I will tell you though is, as a parent, when a new vaccine is introduced, I'm generally not the first to line up. I wait for a year or two to see what happens. I mean, the whole development process, you know, really does, I think, ensure a safe vaccine. But once it goes out into market, then it gets in, what I said I didn't show is what's called phase 4 or post-marketing. It goes into a lot of people. That's sometimes when very rare events are picked up. So one of the things, frankly, you can do is wait. Generally, they're not required for the first year or two. So you can wait for the first year to see, or even the second year to see if anything happens.
I know you didn't want to talk about food allergies, but would you...
It's not that I didn't want to, I didn't have enough time to.
Would you just, I mean, I don't know, but it appears that the food allergies are significantly on the rise. Would you agree with that?
There's no question about it.
And then the second thing is, what do you think is causing this? And what do you think is, shortly, what do you think can be done to stop the rise of food allergies?
I wish if I had a simple answer, I would share it with you. But there is no question that food allergies, and not just food allergies, but serious food allergies are on the rise, particularly in children. And there's a plethora of data to support that. Why that is, I don't have a good answer. However, one theory that I subscribe to a little bit, or more than a little bit, is called the hygiene theory. And that is that human beings have evolved with a whole host of microorganisms. And likely, not likely, almost certainly, many, if not most of those microorganisms are either commensal, which means we live with them comfortably, or actually help us. Some of them certainly cause disease, the ones that I've spoken about today. But the idea of everything being absolutely clean and being germ-free probably is not good for our immune systems, because the general stimulation from all of these microorganisms and antigens that one encounters actually helps to teach the immune system what's important and what's not, and what is dangerous and what's not. And so there was actually a study that I found very heartening. It was published fairly recently, that showed that mothers who licked off their children's pacifiers, those children had fewer food allergies. So as a mother of 4 boys all at once, I was definitely a pacifier licker, and it was very comforting to me. But in all seriousness, what it demonstrates is likely that there is protection from these environmental microorganisms, and eliminating that's probably not a good thing. The other thing is, if you want to induce tolerance, so remember the immune system can either immunize or tolerize, the best way to do that is by eating something. It's called oral tolerance. And so the idea of not giving small children things that you don't want them to be allergic to is counterintuitive to immunology. So, the American Academy of Pediatrics has just reversed itself on not giving children, say, fish and nuts in the first years of life, because obviously don't give them nuts that they're going to choke on, but probably not giving them those antigens by mouth for a long period of time is more likely to induce allergy than it is to protect against it. Though we're just beginning to understand how these signals all interact, and it's very complicated. And that's really, I must give you the caveat, I'm not expert on this. This is just sort of my reading of the literature.
Well, thank you again, Sarah, for your great talk.
Thank you, Barry, and thank you all for your attention.